in the previous class only. Anyway. All right, so we had discussed about the general characteristics of bryophytes. Now today we're going to see the two divisions that the bryophytes come in, that is liverworts and mosses. Talking about the liverworts, they grow in moist, shady habitats such as the banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. These are the habitats wherein the liverworts grow. Now, the plant body of a liverwort is thalloid. Again, thallus-like structure is there. And the example which comes under liverwort is mercantia. Okay, this is like an important example, mercantia. Now, the thallus is dorsiventral and closely oppressed to the substrate. Sort of like it looks like flattened to the surface. Okay, so it was like quickly like this, flattened to the surface. So it looks closely oppressed to the surface or the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Again, no true structures are there, leaf-like, stem-like structures are. Now, the asexual reproduction in liverworts takes place by fragmentation of the thalli or by formation of specialized structures that are known as gamete or gamma cups. Okay. Now, gamma, what are these structures? These are green multicellular asexual buds, just like how conidia asexual spores were there, right? Same way here, asexual buds are there, and those buds are called as gamete. Now these gamete, they develop in small receptacles that are known as gamma cups or the gamma cups. Like they are actually developed inside the gamma cups. Now, moving forward, what happens is that the gamma become detached from the parent body and germinate to form new individual. So it is produced on the parent's body only, but after that it will detach and develop into a new organism. Now, during sexual reproduction, male and female sex organs are produced either on the same or on different thalli. If it will be produced on different thalli, so one will be male and one will be female. Now, the sporophyte is differentiated into a foot, seta, and capsule. This is their body differentiation. Now, you can see, like, instead of being differentiated into roots, stems, and leaves, they are differentiated into foot, seta, and capsule. Now, after the meiosis has been done, the spores are produced within the capsule. Inside the capsule, spores are produced. Then these spores germinate to form the free-living gametophytes. This much is clear. So this is about the liverworts, how their reproduction happens. And the biggest thing to remember over here is the example. The example is mercantia because it is very commonly asked in questions. Okay. So liverworts, the best example is mercantia. Now coming on to mosses. Now, have you people seen those kind of aquariums that are like really huge, and like for those people who make it really extravaganza type, they add many things inside the aquarium, like they would add the water tunnel and whatnot, things are added. And sometimes they also add mountain like things. Have you seen that? Now on those mountains, yeah. And on those mountains, they add you can say greenish grass-like structures. Have you seen this? Wait, I'll show you.
Yeah.
Is my screen visible?
Yes, ma'am.
See, this is how mosses look like. You see? All of these are the pictures of mosses. And they're mostly like, I'll show you moss in terrarium. Actually, terrarium is like, just like how aquarium is there, to show the plants on a land in a jar, terrarium is the structure. See this. And in terrarium, for moisture content and for everything, like look for this picture here. See this green part that looks actually like a grass, but is not grass. That is moss. Okay. Now quickly let us see chlorophyceae. So that, you know, it'll become interesting when we actually see the pictures, because imagine करके नहीं होता है. See, this is how. See, do you remember the kelps that I said? Massive kelp. This is how the kelps look like. Okay, and Chlamydomonas will be the tiniest one out of all. See, this is a volvox. This is to given in your book also, isn't it? All right. And okay, then Pheophyce. See, brown. See, evident brown color is there, isn't it? Evidently brown. And it is brown because of the presence of which pigment? Hmm? Quickly, name the pigment.
Chlorophyll A and chlorophyll C.
Hmm.
Mm,
See
uh,
this, Ali. So beautiful, isn't it?
yeah.
Okay. All right. So now coming on to mosses. Now you people have seen how mosses look like. Now the predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. Now, furthermore, its gametophytic stage has been divided into two stages. The first stage is the protonema stage, which develops from a spore. It is a creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral branch. So, primary protonema, that is the normal first stage protonema, and then comes the leafy stage, which develops from the protonema only. Now, they consist of upright, slender axis-bearing, spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoid. Again, root-like structures. And this stage, the protonema stage or the leafy stage, this stage bears the sex organs. So this is the sexual stage of their life cycle. Moving forward, the vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leafy shoot, like at the tip portion of the leafy shoot. Now, after the fertilization has been done, the zygote develops into a sporophyte consisting of a foot, seta, and capsule. Yet again, the sporophyte in mosses is more elaborate than in liverworts. This line can directly come into question. Like this, they can actually flip it. They'll say that the sporophyte in liverworts are more elaborate than in mosses. And they'll ask you whether the statement is right or wrong, or they'll put it in the assertion reasoning statement or any kind of this statement. So you must remember that the sporophyte in mosses is more elaborate as compared to the liverworts, because we are seeing, see, in the liverworts, there was no further stages, isn't it? But here, protonema and leafy stages are there, which is not there in liverworts. So obviously, it is more, you can say, elaborate. Apart from that, after the sexual reproduction has been done and after the zygote has been formed, then the development takes much more time. And that is also the reason why we say that the sporophyte in mosses is more elaborate. Clear? Now, the capsule contains spores. Spores are formed after meiosis. Now, the mosses have an elaborate mechanism of spore dispersal. Some example of mosses are Funaria, Polytrichum, Sphagnum, right? Peat moss. So, bryophytes is clear. Liverworts and mosses, both of the sections are clear to you.
Yes, ma'am.
Any doubts?
Yeah.
Now we have pteridophytes. Now, pteridophytes, they include horsetails and ferns. Has everyone seen fern? You might have, right? Because it's
Yeah.
the most common kind of plant that we keep at home. See this. Everyone has seen this, isn't it? So this is very
Yeah.
commonly available. And here in India, it's everywhere. So this is the fern and it's ornamental. What is the meaning of ornamental?
Like kept at homes, maybe
But
for
for
like
what?
decoration purpose.
For decoration. Anything that is to beautify or to decorate, that becomes an ornamental plant. And fern is that one ornamental plant. Okay. Now, pteridophytes are used for medicinal purposes and as soil binders. They are frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues. That is xylem and phloem. See, xylem and phloem, you people remember, they are for the conduction of water and food, right? So this
Yeah.
xylem and phloem was not there up until now in any of the phylum or division, right? But until bryophytes, no xylem phloem was there. Now, since slowly, the pteridophytes are moving towards the land condition. Now, pteridophytes are no more aquatic. They're moving towards the land. And them moving towards the land is a very great indicative that now the plant would require certain mechanism in its body so that transportation of water happens, because not now water is not abundant. For that very reason, the pteridophytes, they have the vascular tissues or conductive tissues, that is xylem and phloem. Now the
Ma'am, what does soil
pteridophytes,
binders mean?
soil binders, meaning since they are present on the land, their roots are so deep suited, or like it is so deep rooted that they hold the soil together so that soil erosion does not happen. Okay,
Oh,
it is one of
thank you,
those
ma'am.
plants. Yes. All right. Now the pteridophytes, they are found in cool, damp and shady places, though some may flourish well in sandy soil conditions. Now you may recall that the dominant phase in the life cycle of the is a gametophytic plant. In the bryophytes, the dominant life was gametophytic, isn't it? However, in the case of pteridophyte, the main plant body is a sporophyte. Sporophyte is diploid or haploid?
Haploid.
Sporophyte.
Diploid.
Sporophyte is diploid. Yes. And gametophyte is haploid. Okay. Now, this sporophyte is differentiated. Now, see. Now, since they have vascular tissues present, now they will have true root, true stem, and true leaves. Okay. And these organs possess well differentiated vascular tissues in them. Another name for vascular tissues is conductive tissue. If they ever ask you in terms of conductive tissue, you should know that they are talking about xylem and phloem. Now, the leaves in Pteridophyta are small, microphyls, as in the case of Selaginella, or large macrophylls, as in the case of fern. The word phylls refers to the leaves, and micro, you already know micro means small, macro means huge or large. Now, the sporophytes bear sporangia. Now, this is something new that we are studying. The main plant body, that is sporophytic, it bears sporangia that are subtended by leaf-like appendages called as sporophylls. See, again, phylls means the leaves. Now, the sporangia-bearing leaves would be sporophylls. Okay, just like how the small leaves were microphylls. So, the leaves that would have spores present on it or sporangia present on it, they will be what? Sporophylls. Clear? Now,
Yeah.
in some cases, sporophylls may form distinct compact structures that are known as strobili or cones. This happens in the case of Selaginella and Equisetum. The sporangia produce spores by meiosis in spore mother cell. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophyte, which is known as prothallus. Now see, after the sporophytes, that's a long description they have given, isn't it? How many words have they put in? Inconspicuous, small but multicellular, then free living, photosynthetic, thalloid. 7 characteristics to define the gametophytes of pteridophytes. Okay, so you should know 7 characters are there, right? Now, each of these characters you people understand? Inconspicuous, what does it mean? Okay, first you tell me small but multicellular, meaning of that.
Small shape or small body, but like many cells.
Yes, it is compact, right? Tiny, but has multiple cells. Free living, you understand? Free living?
Yeah, that means they are not attached to something.
Correct. They are not attached. They are not deriving the nutrition from anywhere. They are living just like how we do, right? Normally by themselves. Photosynthetic, they can perform photosynthesis. Thalloid, again, thallus-like structure is there. Gametophyte female. Now, inconspicuous. See, inconspicuous is a word that describes something that is not clear. It's not distinct. Something that is not distinct is inconspicuous. Okay. Now, moving forward. So the gametophytes in the case of Pteridophyta, they are known as prothallus. They have a name. Now these gametophytes, they require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. See, pteridophytes, even though some of their species are widespread, but they are not everywhere. They are restricted to certain areas that will have these conditions, meaning that area has to be cool, that area has to be damp, and has to have shade. Then only they can grow. Apart from that, if the area is there, like all the three conditions are fulfilled, in addition to that, they also require a water source. That water source for their fertilization process. So they have so many, you can say, particulars, right? And this is the reason why that they are not spread to a greater geographical region, okay, because of their need, such a specific need. मतलब इतने नखरे हैं कि ये हर जगह नहीं पाए जाते, इतने नखरे हैं, ठीक है. Now the gametophytes bear male and female sex organs that are known as antheridia and archegonia, just like how it was there in bryophytes. Now, water is required for the transfer of antherozoids, the male gametes released from the antheridia, to the mouth of the archegonia. Then, once that has been done, then the male gamete fuses with the egg, which is present in the archegonia, and forms the zygote. The male and the female gamete will be haploid or diploid?
Haploid.
Always haploid. And the zygote will be haploid or diploid?
Diploid.
Very good. Okay. Now, the zygote thereafter produces a multicellular, well-differentiated sporophyte. See, if the plant body is sporophyte, it will give rise to gametophyte. If the plant body is gametophyte, it will give rise to sporophyte. Because alternation of generation. You remember when we were talking about biological classification, I said something that they can alternate the way their lives have, right? That is alternation of generation, and that is what happens in the case of plants. So zygote, which is produced after the fusion of gametes, it produces a multicellular, well-differentiated sporophyte, yet again, which is the dominant phase of the pteridophyte. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called as homosporous. Homo means same. Sporous is something that bears spores. So homosporous is something that bears same kind of spores. This happens in the case of they have not given the case more in like most of them, okay, but the certain examples that they have set aside, such as in Selaginella and Salvinia, they have different kind of spores. Two spores are there. One will be larger spore, one will be smaller, micro and macro, and they are known as heterosporous. So Selaginella and Salvinia are called as heterosporous. The megaspore and the microspore germinate and give rise to female and male gametophytes, respectively. The female gametophytes in these plants are retained on the parent sporophyte for variable periods. The development of zygote into young embryos takes place within the female gametophyte inside. This event is precursor to the seed habit, considered an important step in evolution. See, like just like how in us humans, if we talk about human evolution, you might have studied chapter evolution in class 12. In that, what did we see? That slowly when like they have connected our evolution along with monkeys, right? So initially we were monkeys, and slowly we developed. And what actually developed? The pattern of living, right? Then cranial capacity developed. We then after evolution was done, the present humans could speak. They had intellect, right? So that is the evolution for humans. Same way, if we are talking about the evolution in terms of plants, then what happens is that they did not used to have seed. In them, seed formation, flower or fruit formation, these things are considered as a very advanced thing. So in pteridophytes, since the zygote was developing inside the female gametophyte, right? Inside. That is sort of like the scientists saw that, okay, this is something relating to the seed, like because seed is also found inside the fruit, right? So this development of zygote into young embryos, which takes place within the female gametophyte, this is considered as like a precursor to seed habit, meaning something that will give rise to seed habit in the near future. Clear? Now, the pteridophytes are further classified into 4 classes. Psilopsida, under which comes Psilotum. Lycopsida, under which comes Selaginella and Lycopodium. Sphenopsida, under which comes Equisetum. Pteropsida, under which comes Bryopteris, Pteris, Atrianthus. Again, very important. All of these classes are extremely important. Like which organism belongs to which class, that is important. So, pteridophytes is over. Is it clear? Any doubts?
No, no.
All right. Now moving on to gymnosperms. Now gymnosperms, they are the ones that have the seed. They produce seeds. The first seed producing plants are gymnosperms. But what is unique about their seed is that just like, for example, let's just take an example of mango. If you look at mango, then the seed of the mango is present inside, but it is covered by the flesh that we eat, and that flesh is also covered by the chilka, the peel, isn't it? So you take out the peel, you take out the flesh of the mango, and then there is the seed of the mango, right? So that seed is enclosed within a fruit, isn't it? But for the case of gymnosperm, the seed is not enclosed within a fruit, so their seed is known as naked seed. It's not covered by anything. So gymnos means naked. Sperma is referring to the seed. So these are the plants in which, and see, okay, do you people know ovule and ovary of plants? So the
Yes, ma'am.
ovule, yeah, okay. See, the ovary that we talk about, right, in which the egg cell is there, isn't it? That ovary develops into a fruit in the case of plant, and the ovules which are present inside the ovary, they develop into the fruit, seed. Okay. So just like how seed is present inside the fruit, same way ovules are present inside the ovary. So in this case, in the case of gymnosperm, the ovules, they are not enclosed by any ovary. Okay, no ovary wall is there and they are exposed both before and after fertilization. Now, the seed that develops post fertilization, they are not covered, meaning they are naked. Now, gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza. The fungus, along with the roots of higher plants, that is mycorrhiza. And that fungus forms the association with pinus. Pinus is one species of gymnosperm. Okay. Have to find this. This is the pinus tree. Okay. Again, if you have ever visited hilly areas, they are mostly seen there also. And what is another thing? Sequoia. That's not sequoia. Oh, Sequoi. The giant redwood tree, one of the tallest tree species. See, evidently redwood is there, right? So this is again a gymnosperm. Now you see how the plant's height has increased and it has become a full grown tree, right? Now, the seeds produced by this are not covered by the ovule. So mycorrhizal association is there in the case of Pinus, while in some others, that is in the case of Cycas, small specialized roots that are known as poroloid roots are associated with nitrogen fixing cyanobacteria. Now the roots of Cycas have cyanobacteria present in them that are responsible for nitrogen fixation. The stems are unbranched in the case of Cycas or branched in the case of Pinus and Cedrus. The leaves may be simple or compound. In Cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremities of the temperature, such as of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce the water loss. Now see, when we saw the picture of Pinus, if we see it closely, do you see how the leaves are spike-like? Isn't it? Spikes are like normally when we draw the leaves, we draw it like this, right?
Yeah.
This is the representation of leaves in our mind, isn't it? But this
Yeah.
is the leaves of angiosperms. But in the case of Pinus, the leaves, if you look at here, they are not like the traditional leaves that we draw, isn't it? It's like so different. Even if they give an outline of that kind of leaf, but if we see it, like if we zoom it or if we see it very closely, we'll find that the whole entire thing is made up of spikes. There is a reason why this is so. See, spikes. Why? So as to reduce the water loss. When the leaf surface will be reduced, then the water loss will also be reduced. Yeah. So this is the
Yeah.
reason why, yeah, they have spike-like structures or conifers, needle-like leaves are there. Now, the
Is it
gym.
like cactus?
Mm,
Because even cactus
no.
have a weird
See,
leaf,
yeah.
right?
See, cactuses are like they have a proper fleshy stem, and on that they have thorns, isn't it? There are thorns present. But in this case, see, cactuses do not have leaves at all. Their leaves are so reduced that they have become thorns, isn't
Yeah.
it? They do not have leaves at all. This is the stem part of cactus. But in this case, leaves are there. These are the leaves only, but their leaves are needle-like. Okay?
Yes, ma'am.
Now, the gymnosperms are heterosporous, meaning they produce two kinds of spores, that is, microspores and megaspores. Again, the spores will be haploid. Now, the two kinds of spores are produced within the sporangia that are borne on the sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. Same way, the compact structures, the strobili, here is also known as cones. Now, the strobili bearing microsporophylls and microsporangia are the male strobili. It's seen here also in this picture. See, okay, you might have heard about pine cones. Pine cones? Where does that come from? It comes from Pinus only, pine cones. Those are the sporophylls or the strobili only. See this compact structure, the brown structure you're seeing right in front of you?
Yeah.
This reduced structure, yeah, that is the strobili. See, this is the strobili. See this thing. Clear? This is how, so
Yeah.
just imagine them being compactly into form this kind of a structure. That is a cone or a strobili. It doesn't look like a cone, but anyway, if they are saying it is a cone, then it is a cone. So the ones that would bear the microsporophylls and the microsporangia, they are the male strobili or microsporangiate. The microspore develops into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is now called a pollen grain. Their gametophyte is so reduced that it becomes a pollen grain. It is not evident, just like how gametophytes were a dominant phase in the body of bryophytes and pteridophytes, isn't it? Like it was a phase in their life. But here the gametophytes have been reduced. Now, अब ये चक्कर ही खत्म हो रहा है. ठीक है? Now they are being confirmed to pollen grains. Here. Now, the development of pollen grains take place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia, they are known as megasporangiate or female strobili. Let us search that also, male and female strobili. See this. You see? Structure. Right?
Yeah.
Again, strobili, the cone. This is what it is. Okay, you might have seen it also. And you might have seen for those people that pine cones. It's like very famous. I think in the time of Christmas or something, people use it as decoration.
Yeah, they put it on the Christmas trees.
Yeah. Those pine cones are nothing but your gymnosperm. So now you know more than
Yeah.
isn't it? Okay. All right. The male or female cones or strobili may be born on the same tree in the case of pinus. However, in cycas, male cones and megasporophylls are born on different trees. It can be born on the same or different. Now, the megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called as the ovule. Meaning the ovule, just like how it in angiosperms also it is surrounded by nucellus, here also. Now the ovules are borne on the megasporophylls, which may be clustered to form the female cones. The megaspore mother cell divides by meiosis to form 4 megaspores. One of the megaspore enclosed within the megasporangium. See, the basic thing here to understand is that the microspores are present inside the microsporangium. The megaspores are present inside the megasporangium. And when this megasporangium would develop into a multicellular female gametophyte that will have the archegonia or the female sex organ, meaning going back to the adult stage. Now the multicellular female gametophyte is also retained within the megasporangium. Clear? Now, moving forward, unlike bryophytes and pteridophytes, in gymnosperms, the male and female gametophytes do not have an independent free living existence. They remain within the sporangia retained on the sporophyte. So here, they are not free living, they are present on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on the megasporophylls. The pollen tube carrying the male gametes grows towards the archegonia in the ovules and discharge their content near the mouth of the archegonia. Then, after the fertilization has been done, the zygote is formed and then the zygote develops into an embryo. And then the ovules will develop into the seed and these seed will not be covered. This is how this gymnosperm is done. And some of the species, they have not given species at the end, but they have given in the diagram. So Cycas, Pinus, and Cedrus, these things we have studied in the text only. There is the giant redwood tree, Sequoia, and this ginkgo or ginkgo, whatever people call it, ginkgo biloba. See, it is very famous for having such huge leaves, fan-like leaves. Okay, so ginkgo biloba is again very important to remember. Clear? Gymnosperm is clear? They are like, gymnosperm is my least favorite division out of all of them. I would prefer studying about algae, bryophytes, but I hate it. All right, now we are on to the best one out of all of them, that is angiosperm. It's the best.
Ma'am, angiosperm and plant life cycle is deleted, right?
Angiosperm and plant life cycle is deleted.
Yes.
Is it for NEET also?
It's for NEET also, because I've just searched.
Okay, okay. Good. Even better. So, okay, then with this, we have completed the plant kingdom chapter. Now tell me, you people, if you have any doubts, anything that you want to understand. So what is the next chapter that I was going to do? Was, according to you, was this chapter nice or biological classification? Which one? Or both were
Biological classification was better.
better than this?
Yeah.
Only plants is like boring, isn't it?
Yeah.
Do you know what is my major? Botany.
Plants.
Botany. Okay. Let's see. The chapters that I've assigned to you for this period of time, we have completed biological classification and plant kingdom, right? Now, I'll give you a choice to choose one chapter out of the three I'm saying. Okay, it's your wish. See, I have morphology in flowering plants, anatomy in flowering plants, or cell the unit of life. Which one do you want to do? Next. The choice is yours. Do you want to do cell so that change of pace is there? है थोड़ा सा plan से हट जाए. Fine with you people? Because even I am bored. That's why. So I'll start with cell. Okay. Just give me one time. Okay. And as far as this plant kingdom goes, no matter if you're bored or not, do you have any doubts? Is there anything that you want me to teach again? The whole chapter is
No.
clear?
Yes, ma'am.
Okay. How's your physics and chemistry preparation going? 11th physics is, how do you find physics? Like which is your strongest subject out of the three?
Bio.
Bio, both of you.
Yes, ma'am, bio.
And what is the condition of physics and chemistry?
For me, chemistry is handleable, and I can handle it. Physics, I don't know.
With physics out of hand.
No, I cannot.
But practice as much as you people can, okay? All the numericals and everything, practice as much as you can, because the applications of the questions is what is the most important thing, right? And yeah. See, bio, I'm taking full responsibility from my side. Physics, chemistry, I don't. Bio, I take full responsibility. Like up until now, whatever I've taught, it goes into your brain, right?
Yeah. Yes, ma'am.
Sure.
Sure, sure.
When the things do not go into your brain, kindly inform me. I'll try a different method to put it inside your brain. I will put it inside your brain, but I'll try a different. Okay.
Yes, ma'am.
Yeah. Okay. I wish I had you people for like normal classes. Like you would have studied the normal 11th with me, then normal 12th with me, then because right now it is so concise, the time period is also very short, so I can give you concise information only, that the information that is important for NEET, right? But we would have had really like a fun time when you would study normally. Anyway, so let us start with cell, the unit of life. Screen is visible to you people?
Yes, ma'am.
Now, cell being called as the unit of life, now we both know that it is so important, right? None of us would be living or considered as living if we did not have cell in our body. And for as far as the unicellular organisms go, Even if one cell is present in their whole body, still that makes them living, right? Now, all organisms are composed of cells. Some are composed of a single cell. We call them unicellular. Some are composed of more than one cell, like many multiple cells. We call them multicellular organisms. Now, unicellular organisms are capable of the first thing, independent existence. They can live independently. Secondly, they can perform, even after being just made up of one cell, they can perform all the essential functions of life, right? So these two properties are the properties of a cell. What can constitute as a cell? Something that can exist independently and something that can perform all the essential things that are there for life, then that is a cell. Now, anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental, structural, and functional unit of all the living organisms. Anton von Leeuwenhoek first saw and described a live cell, a living cell. Robert Brown later on discovered the nucleus. Then the invention of the microscope and its improvement leading to electron microscope revealed all the structural details of the cell. See, slowly, when the science was like evolved, then electron microscope came, and when, see, electron microscope is so highly precise that it can show such a deeper level, right? We go and see the cell. Now, talking about the cell, first of all, these two names are very important. Like they'll be asked forever. Like even if you're done with studying 11th also, then also it will be there. Anton von Leeuwenhoek, he was the person who described a living cell. Then Robert Brown, he described the this thing, nucleus. He discovered the nucleus. Clear? Now, cell theory. Now, in the year 1838, Matthias Kleiden, who was a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant, right? Multiple various kinds of cells are coming together to form the tissue. Okay, do you people know this? Cell is there. Then a group of cell is known as tissue, right? A group of tissue is known as a group of tissue, like a group of cell makes tissue. Then a group of tissue would come together and make
Organ.
organ. Then a group of organ would come together and make
Organ system.
very good. Right? We'll have fun when we study animal kingdom. You people will enjoy it thoroughly. I'll make it so easy for you. Okay. Another thing I always like to ask this to my students is like, in class 11th, which is the toughest chapter in biology, according to you? Toughest, like you despise that chapter, you never want to study that chapter.
Oh, those I have those 3 chapters: the
Three?
photosynthesis and the cell chapters with all the cycles and everything.
Huh?
Like the big cycles.
Oh, okay. Photosynthesis,
remember.
respiration.
Yeah. I had that somewhere.
3, I asked for 1, you're giving me 3?
Like I don't like those big chapters.
I'll make you love it, don't worry, that's my duty.
Okay.
And yes, you tell me.
Ma'am, the chapter which consists of Krebs cycle. I don't know that.
Respiration.
Respiration.
Yeah.
It's
Krebs
one
cycle,
common
Calvin
chapter
cycle,
that
those cycles.
everyone hates that. It's okay, don't worry, I'll make you love it. I think you as far as I think, you people did not used to like biological classification also initially, because many students hate it, since it is like, again, not so relatable, isn't it? Is that the case with you people also?
No, that chapter is fine. Like,
Hmm.
but these chapters are just not.
Which one? Respiration.
Yeah.
Yeah, see, there are like a lot of chemical technicalities in
Yeah,
that
that's what.
chapter, isn't it? Anyway, we'll deal with it when we come to it. Good. for me to know that कहाँ पे ज़्यादा मेहनत करनी पड़ेगी. All right. Okay. So talking about the cell theory. So Matthias Kleiden, and he was a German botanist. So he examined all the plants. Now he said that when he was examining the plant, he's seeing that certain regions of the plant have different kinds of cells. The other ones have different kinds of cells. So they are made up of multiple various kinds of cells. At the same time, when Kleiden was doing his discovery, at the same time there was Theodore Swan. He was a zoologist, a British zoologist, and he studied different types of animal cells. And he reported that the cells had a thin outer layer, which is now known as the plasma membrane. So, meaning the cells that are present, they are surrounded by one membrane, which is known as plasma. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells, which is 100% absent in animal cells. There is no animal that has cell wall cells, but cell wall is there in plants. Now, on the basis of this, Swan proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. The different kinds of cells are only made from the initial cell zone, right? Now, then they came together and they collaborated. And then they said that let's formulate a cell theory in which what they did, like Kleiden and Swan together, they said that firstly, the cells are made up of all the living cells are made up of cells, like all the living organisms are made up of cells, and the cells certain make their own kinds into their products, right? The cells give rise to new cells. Now, they told about how different kinds of cells exist in different kinds of organisms. Then came another person whose name was Rudolf Virchow. In the year 1855, he said that, now he talked about how the cells are formed. Okay, he talked about the formation, and he explained that the cells divided, like cells form new cells by division, and new cells are formed from the pre-existing cells. The cells that have been existing for a while, those cells give rise to the new cells. And this phenomenon is known as omnis cellula e cellula. Okay, this is the phenomena that pre-existing cells give rise to the new cells. That is omnis cellula e cellula. He modified the hypothesis of Kleiden and Swan to give the cell theory a final shape. Now, what we have we have two proponents in the cell theory. First is that all the living organisms are composed of cells and products of cells. Secondly, all the cells arise from the pre-existing cell. So that is your cell theory, which was given by Kleiden and Swan. Even though Rudolf Virchow gave such a huge contribution to their theory, his name is nowhere seen. Whenever you will hear cell theory, you will hear Kleiden and Swan. Now, one thing to remember here is who out of them, which one out of them is British, which one out of them is German. Because, you know, like in assertion reason, they might give it like Matthias Kleiden, a British botanist, right? Then this question becomes, like the whole sentence becomes wrong, isn't it? Because he is in fact a botanist, but he is not British, he is German. So they maybe can twist the sentence like this. That is why I say that whenever you study keeping NEET in mind, you like just like how I am teaching you, I'm underlining every single word, every single line. This is how you're supposed to study also. You cannot skip one line, you cannot skip even one word. Okay? Because this NCERT, abhi, this is March, right? March, April, and in May you have your exam. So for the whole of April and the rest of the March, you make this book your everything. Sleep with it. Okay, you put it near your pillow, sleep with it, so that whenever tension hits, you open it, you read quickly. Because I'm telling you, everything that is going to come from here. Every line is important, every diagram is important. I mean, make your what do you say, preparation so good कि अगर मैं कोई भी एक ऐसे पैराग्राफ बोलूं, so you should remember, okay, it is from that chapter, from this page, यहाँ पे ऐसा ऐसा बना हुआ है, इतना अच्छा कर लेना है ना. Because in NEET 360 मार्क्स तो हमें यहीं से मिल जा रहा है, हमारे फेवरेट सब्जेक्ट से, है ना. बायोलॉजी इज आवर फेवरेट सब्जेक्ट, है ना. मैं तो पढ़ाती हूँ, तुम पढ़ने आए हो, तो दिस इज आवर फेवरेट सब्जेक्ट. तो हम इसमें तो कोई चूक रहने ही नहीं देंगे. We cannot let any question go wrong in this particular. Okay. All right. Chalo, then next. Okay, and see what I was thinking after I'm done with each and every chapter, I'll provide you with the previous year questions that came from this particular chapter, है ना? So you attempt that without cheating, without seeing the answer, and then कहीं पे भी doubt होगा, कहाँ पे कैसे कौन सी thinking लगनी क्या लगना, you ask. Okay? All right.
Okay.
Now an overview of cell. Now you have earlier observed cells in an onion peel, no, or human cheek, no, under the microscope. We haven't, but still, चलो बोल रहे हैं तो. Now, the onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it, it has cell membrane or plasma membrane. The cells of the human cheek have the plasma membrane, that is it. It would not have the cell wall. Inside each cell is a dense membrane-bound structure that is known as the nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material, that is the DNA. Cells that have membrane-bound nuclei are known as eukaryotic cells, whereas the ones that do not are known as prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix, which is known as the cytoplasm, occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plants and animal cells. Anything that happens inside the cell happens inside the cytoplasm. Okay, so just imagine cytoplasm like the living room of your house. We can live in separate rooms, but whenever the guests arrive or whenever anything, like whenever you enter into the house, the first room that you come into is the living room only, isn't it? That's where people gather. Yeah, so cytoplasm is the living room of the cell. It is where every kind of drama happens. Okay, now various chemical reactions occur inside the cytoplasm only to keep the cell in the living state, because the cell cannot stay still. If the cell is still, then the cell is dead. So the cell has to constantly do something. It can't sit idle. Okay. Now, besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures that are known as organelles. Now, what are those? Now, see, to remember which organelle has the membrane, which one does not, is very important. Okay, now they are referring to the membrane-bound organelles. So, endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. All of them, just one second. All of them have membranes surrounding them. They may have one or two membranes may be there also, but you must remember that these are the organelles that have membranes surrounding them. These are the membrane-bound organelles. Now, the prokaryotic cells, they lack such membrane-bound organelles. They are not present at all. But there is one organelle, only one, that is ribosome, which is a non-membrane-bound organelle. It does not have any membrane, and it is present in both eukaryotes and prokaryotes. Ribosome does not have any membrane surrounding it, and it is found in both. Within the cell, ribosomes are not found not only in the cytoplasm, but also associated with two organelles. One is chloroplast in the case of plants, and mitochondria, and on the rough endoplasmic reticulum. So ribosomes, apart from being the cytoplasmic ribosome, see, the ribosomes that are normally found in the cell, we call them as cytoplasmic ribosomes because they are present in the cytoplasm. But apart from that, they are also present in some organelles such as chloroplast, mitochondria, and rough endoplasmic reticulum. And remember one thing, that the chloroplast will not be present in animals, right? And chloroplast contains what?
Chlorophyll.
Chlorophyll.
Chlorophyll. Now, animal cells contain another non-membrane bound organelle which is known as centrosome, which helps in the cell division. Centrosome. We will talk about it in depth when we talk about cell cycle. Now, cells differ greatly in size, shape, and activities. For example, mycoplasma. You remember mycoplasma? Right? These are the smallest cells. They are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. The largest cell is the egg of the ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells, not the largest, but the longest. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary within the with the function that they perform. Clear? So this is about how the cells are seen. Mesophyll cells that are found in the leaves, they are round and oval. This is tracheids again found in the plants, such an elongated tapering. Then this is the longest cell, that is nerve cell. Red blood cells, round and biconcave structure they have. White blood cells are amoeba-like, irregular. Then columnar, column-like, isn't it? All right. Now we'll talk about prokaryotic cells. Now the prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cell. They may vary greatly in shape and size. The 4 basic shapes of bacteria are bacillus, coccus, vibrio, and spirillum, right? What we have discussed already. Now, the organization of prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. Now, all the prokaryotes would have a cell wall surrounding the plasma membrane, except for mycoplasma. We remember that they are wall-less, right? Now, the fluid matrix is the cytoplasm. No well-defined nucleus is there. See, nucleus is the structure in which the chromosomes are there, meaning our genetic material is there. Now, if this nucleus is actually like distinctive in us, like in eukaryotes, why? Because this wall is there. If we remove this wall, then the chromosome will be lying in the cell. If this is the cell, what happens? See, the chromosome will be lying in the cell only. Okay. Is my screen working towards your side? Yeah. So if this is the cell, now this was the nuclear membrane. If we are talking about the prokaryotic cell, if we remove the nuclear membrane, the genetic material will be lying in the cell only. So here, if anything, see in biology, if anything is not covered by a structure, we'll call that thing as naked. Just like how naked seed was there, here naked genetic material, because it does not have any envelope. Now, the genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA is the one that that refers to the nuclear DNA. Okay, genomic. Apart from the genomic DNA, which is single chromosome and circular DNA, many bacteria have small circular DNA outside the genomic DNA, meaning they have extra DNA. Those smaller DNA are known as plasmids. You might have studied about plasmid in biotechnology. You remember cloning vector? So those plasmids are extra chromosomal DNA. Remember, extra chromosomal DNA. That is apart from the genomic DNA. And this plasmid is very important for the prokaryotes, mostly the bacteria. Why? Because they offer certain unique phenotypic characters to such bacteria, such as antibiotic resistance. The antibiotic resistance comes from the plasmid. Phenotypic, you understand? This also you must have studied in that genetics chapter. Phenotype, genotype. What is phenotype?
Phenotype is the external appearance.
And genotype is the
It's the genetic makeup.
Very good. Now, one such character that is offered by the plasmid is resistance to antibiotics. Now, they are saying that in higher class, you will study about that. Okay. Now, nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotes except for ribosome. Ribosome is common in both. The type of ribosome is different. For example, for prokaryote, it is 70S. For eukaryotes, it is 80S. Prokaryotes have something unique in the form of inclusion. Inclusion bodies are present in them. A specialized differentiated form of cell membrane is known as mesosome. It is the characteristic feature of prokaryote. See, if we talk about the plasma membrane of prokaryote, if it is like this. Okay, now mesosomes are found or formed from the invagination of the plasma membrane. Meaning, if the plasma membrane is like this, then the mesosome is formed by the invagination. Invagination means deepening of the plasma membrane to form a vesicle-like structure, that is mesosome. Okay, see, these are the infoldings of the cell membrane, or you can also say that invagination of cell membrane of plasma. Now, this much is clear. The intro part of prokaryotic cell is clear.
Yes, ma'am.
Okay, good. So now, cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound 3-layered structures. So one cell envelope is made up of 3 layers. Okay, let us see what are those layers. So the outermost layer is glycocalyx, then there is cell wall, and lastly there is plasma membrane. So glycocalyx, cell wall, plasma membrane. 3 layers when together make up one cell envelope. Although each layer of the envelope performs this distinct function, they act together as a single protective unit. Bacteria can be classified into 2 groups on the basis of differences in the cell envelopes and in the manner in which they respond to the staining procedure developed by Christian Gram, the person, which is those that take up the Gram stain, that those bacteria that get stained when you stain them, those are known as Gram-positive bacteria, and the ones that do not get stained, they are known as the Gram-negative bacteria. And their staining ability, like whether they get stained or not, it depends upon the cell envelope, the composition of the cell envelope. Okay. Now, glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath that is known as the slime layer in some, while in others it may be thick and tough called as the capsule. So this glycocalyx only is of 2 types. It can either be loose called as the slime layer in some of the bacteria, and it can be thick and strong as in called as the capsule present in some other bacteria. So different bacteria can have different type of glycocalyx depending upon the composition, again what it is made up of. Now, the cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The cell wall is like, this is the function. It provides shape, like a proper shape, and it also provides a structural support, and it does not allow the bacteria to either burst or to collapse. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of eukaryotes. Now, a special membranous structure that is known as the mesosome, which is formed by the extension of the plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. See, just like how I drew it like this is a vesicle. But if it is present like this, then that would become a tubule. Same goes for lamella when it will be present in such a way. Okay, so they help in cell wall formation. Now see, the function of mesosomes, here are they. Cell wall formation is the first function. Then DNA replication. Okay, cell wall formation, DNA replication, and distribution to the daughter cell. Apart from that, help in respiration, excretion, to increase the surface area, and the enzymatic control. So the mesosomes are very important for prokaryotes as they perform these many functions. Now, in some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm which are known as chromatophores. See, again, the membrane extension which contains the pigment will be known as chromatophores. They will also have other functions, but they will be extensions of the membrane only. We are talking about the extension of the plasma membrane. Clear? Any doubts up till now?
No, ma'am.
Now, bacterial cells may be motile or non-motile. If they are motile, they would have the filamentous extension from their cell wall that is known as flagella. Now, one more thing to remember here, that flagella in the case of prokaryotes, it is an extension of the cell wall, not of the plasma, right? They can confuse you. They can say that filamentous extension from their plasma membrane is known as flagella. Now, what usually happens, you'll see flagella, you'll see filamentous extension, you say, okay, right, but it's not, right? It is the extension of cell wall, not plasma. Now, bacteria show range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. This is the components of one flagella. The filament is the longest portion and extends from the cell surface to the outside. So if this is a cell wall, so it will start from here, but will extend outside. Okay. Now, apart from flagella, besides flagella, pili and fimbriae are also surface structures of the bacteria, but do not play a role in motility. No movement. Pili has another function, fimbriae has another function, but none of them are involved in the movement process. The pili are elongated tubular structures that are made up of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. See, for example, if this is the cell of bacteria, bristles will be like this. This is fimbriae. Okay, so it will help the bacteria to attach to any surface. This much is clear?
Ma'am, fimbriae were also present in the fallopian tube, right? That were for the movement of, no, for capturing the ovum.
Correct. That's there. But again, a different composition in us humans. But again, it does not make the cell motile, isn't it? It's responsible for, collection of the the ovule, the egg, but does not help in any locomotion process. Now, moving forward, we have ribosomes and inclusion bodies. See, since the prokaryotes are so primitive, so they have limited structures that are present in them. In prokaryotes, ribosomes are associated with plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S. Don't add them, you'll never get 70. But if you look at the diagram of ribosome, okay, ribosome sort of has a larger subunit like this and a smaller subunit like this. Okay, ribosome is made up of two subunits and both of them are joined together with the magnesium dipositive ion, the concentration of it. Okay. Now this larger subunit, they are saying it is of 50S and the smaller subunit is of 30S. Now this S, okay, this is the sedimentation coefficient. Okay, it has nothing to do, it shows that at which rate will it settle down, because the larger would settle down faster as compared to the smaller one, and altogether it makes the 70S ribosome of prokaryotes. Okay, now when present together form 70S prokaryotic ribosome. Ribosome are the site of protein synthesis. In the case of prokaryotes also, in the case of eukaryotes also, ribosome is equal to synthesis of protein. Okay. Now, several ribosomes may attach to a single mRNA and form a chain that is known as polyribosome or polysome. This is mRNA like this. Many ribosomes will be attached onto it like this. Okay, this structure that you're seeing, and this will be called as the polysome or polyribosome. Many ribosomes attached onto one mRNA. Okay, chain, one chain. So this is about the ribosome. Now we have the inclusion bodies. Now the reserved material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. They are there for the storage of reserved food material. They are not bound by any membrane system and they lie free in the cytoplasm. Does not have any membrane surrounding them. So they are freely there in the cytoplasm. Examples of inclusion bodies: phosphate granules, cyanophycean granules, glycogen granules. These are the reserve material storing area. Now, gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Gas vacuoles are there. Okay. So, these are all the structures that are present in the prokaryotic cell, amongst which we have. Okay. The cell envelope consists of 3 layers. Name them from out to inside. Outermost is
Glycocalyx.
and then inner to glycocalyx.
Cell wall, plasma membrane.
The ribosome of prokaryotes is of which type? Which type? Tell me. Ribosome of prokaryotes is of which type? 70S. Okay. Is like okay. Okay. Is the ribosome membrane bound or not? In the case of prokaryotes. The ribosome of prokaryotes, is it membrane bound or not? Not membrane bound. Then what about the ribosomes of eukaryotes? Membrane bound or not? Membrane bound. Eh? Membrane bound. This is what I said initially. The ribosome is the only organelle. Whether it is present in prokaryotes, whether it is present in eukaryotes, it will be non-membrane bound, does not have any membrane. Okay. It will only differ with respect to the sedimentation coefficient. 70S in prokaryotes, 80S in eukaryotes. Okay. Always remember this one. So with this, we have completed this thing, prokaryotic cell. Moving forward, we have eukaryotic cell. Now, eukaryotes include all the protists, plants, animals, fungi. All of them are eukaryotic organisms. Now, in this particular type of cell, there is a compartmentalization, meaning the cell has been divided into certain compartments of the cytoplasm, because all of the organs, most of the organs that are present, the organelles, they are membrane bound, surrounded by a membrane, right? Because of which it creates a separation between each of them. Eukaryotic cell possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. See, to commonly understand the chromosome, you people understand DNA? The genetic material that we have, that is DNA, right? Now, that DNA is carried on a chromosome. So, you can call chromosome as the physical carriers of DNA. These are the structures that are the physical carriers of DNA, which is our genetic material. Now, all eukaryotic cells are not identical. Plant and animal cells are different, as the plant cell possess the cell wall, it will have plastid, and it will have a large central vacuole. three things that are there in plant cells that are not present in animal cells are cell wall, plastid, and large central vacuole. Vacuole is almost negligible in the case of animals, but some do have. So a large central vacuole, meaning a big vacuole present right in the middle of the cell, is not there in the case of animals. It is there for plants. So three things: cell wall, plastid, and what is the last one?
Large central vacuum.
Large central vacuole. These things are not present in animals but are present in plants. On the other hand, animal cells have centrioles that are absent in almost all plants. So animals also have something that is not there in plant cell, and that is centriole. Centriole is membrane bound or non-membrane bound? With this, I would understand that you people have been listening to me. Centrioles are membrane bound or non-membrane bound? Membrane bound or non-membrane bound?
Membrane bound.
It is non-membrane bound. Centrioles are non-membrane bound. Okay. All right. So the intro to the eukaryotic cell is clear. Any doubts?
Yes, ma'am.
No doubts?
No.
Sure.
Yeah.
Ribosome in prokaryotes is membrane bound or non-membrane bound?
Non-membrane bound.
Ribosomes in eukaryotes?
Non-membrane bound.
Okay. Centrioles are present in animals and plants both, or in just animals?
Only in animals.
Only in
Animals.
animals. Clear na? Okay. All right. ठीक है चलो इतना this much is clear. पक्का sure?
Yes, ma'am.
So now we have the whole thing to discuss, each of the cell organelles and the structure, but I will start this in the next class. Okay, because अब मेरी अफ्तारी की time हो रही है, अब इससे ज़्यादा मैं नहीं बोल पाऊंगी. So this is it for today's class. Have you people are you people done with your aftar? No.
No, ma'am.
How much time is
It's
left?
3:30 here.
Oh, it's 3:30 there, and
Yeah, it's 3:30 here.
and iftar happens at
5:50. 5:52.
5:50, yeah.
okay, it's 5:59 here. So it's almost my time. Okay, so let's just keep it till here only. Now in the next class, we'll start with cell membrane. Now listen, biological classification is done, plant kingdom is done, and quarter of cell is done. इसे prepare करते हो. अब no excuses. अब थोड़ा strictly है ना. In the next class, I will ask. आज भूल गई थी, ठीक है? अगली class नहीं भूलूंगी. Okay? So I will ask question. ये तीन chapters एकदम मतलब दो chapters और इसका कितना part आज पढ़ा है. It should be completed. मैं बता रही हूँ जैसे जैसे आई एम कंप्लीटिंग द क्लास यू कंप्लीटेड साइड बाय साइड, तभी फायदा होगा, नहीं तो फिर नीट आ जाएगा और फिर सब बैकलॉग में रहेगा. ठीक है, वी कांट अफोर्ड दैट, है ना, टाइम बहुत कम है. पहला चैप्टर नॉट पहला चैप्टर, बायोलॉजिकल क्लासिफिकेशन एंड प्लांट किंगडम लिख लो. बायोलॉजिकल क्लासिफिकेशन, प्लांट किंगडम एंड अप टिल हियर यू पीपल विल हैव टू प्रिपेयर, है ना, एकदम अच्छे से याद करके आओगी. आई विल आस्क क्वेश्चन. ठीक है, पक्का से आई विल आस्क क्वेश्चन. सो